Okay, we're recording. Did anybody actually end up using the recording from last week? Did that work for anyone?
It is very helpful.
Okay, great. Okay, so if we've got OBS Visual and Virtual Cam installed, that's great. If not, we can work on that later. And then I think what I'd like to do is just start with trying to work through some bash exercises. So for this, does everybody have a sort of bash terminal that they can use? Or does anyone not have a bash terminal? Okay. Do you have one, Batya?
I do. I have several.
Okay,
I
great.
get myself confused because there's too many.
Okay, well, so if you guys were able to install Ubuntu, like I'm pulling up right now, then you already have it installed. Most Macs have it. I think it's just the normal terminal. We might run into some issues, and if that is the case, then we'll just skip this section and make sure that you guys have the right packages installed and whatnot and go from there. So I'm using the Linux subsystem, just out of question. No, okay, why don't we come back to this and we'll first do the installation of QIIME, because that will make sure that we're all on the same page. So I'll just jump down to that section. And we'll come back to bash afterwards. I think that'll make more sense anyway. So QIIME, we're going to be, does anybody not have it installed?
I don't, but my plan was to use it on the lab computer, the Mac. Marilu, do we have it on? Do we have QIIME2 already on the lab computer?
Muted. Sorry. We don't have it. We need to install it. I could install it once I'm there if you like.
Otherwise, I can next week sometime. Up to you.
Yeah.
Figured that would be easier than my Windows laptop that doesn't have much processing power.
Okay, so I see some people were not able to figure it out, and so why don't we just work on making sure that we can all do this. So installing QIIME is fairly simple on a Mac. If you're on a Mac, why don't you go to the Conda, this website here, and you're going to install Conda onto your computer. You can use this code wget to download the latest version in terminal if you want, or you can download it in another location. And then you're essentially just going to run this code. Okay, so why don't I just walk us through that? Out of curiosity, who here is all running on a Mac? I think you should be able to use the reaction button too if you don't have a video, and you can put a thumbs up. Okay, so we got some wealthy people out here. Okay, well, let's just go ahead and do it then. I have never installed anything on a Mac in terms of QIIME2, but it shouldn't be that difficult. So we'll go to this conda.io package. You can open up your terminal. I'm going to put the link in the chat here. So what I would do is download the Miniconda installer. You do not need the whole package. Miniconda is smaller. So you can, I think you should be able to maybe even download this as it's, yeah, here you go. It's a Python version 3.6. This is the Windows, so right here. So you can do it by clicking, or you can just open up bash, or your terminal in this case, and run this command. No, this is, I'm sorry, this is once you've actually downloaded the file.
Yeah.
Yeah, so you'll want to go here and just download this. Or you can do this command right here, wget, if if you have wget. So for that, you would just simply just put that in your terminal, and you can see for me it will automatically start downloading it. It's not a very large file. It's not a large file, and then all you need to do is run the command that's after that, which is bash Miniconda latest. Now, if I do that on my system, I'm sure I'm going to have an issue because it's for macOS specifically. So I won't do it, but it's as simple as just running those two commands, accepting the defaults, and then close and restart the terminal. So once you run that command, you just run that afterwards. And then same thing, like who's running on a Windows system, Windows 10, you can raise your hand. Okay, excellent. So for you, Batia, you're going to want to, okay, Mike is running it too. What you're going to want to do is install the Linux subsystem. So why don't we look at that code? So that's as simple as just pressing the Windows key and searching for Windows features. Windows features.
So I have that checked off already, but I can't seem to find it anywhere. And I've already restarted my computer multiple times.
That's good. So you just check that off. Make sure that this is checked. Mike, is it checked for you? If not, it's really simple. You just press the button.
I was muted. I just checked it.
Okay. Then you should press OK. And then for Batia, you're going to go into the Windows or Microsoft Store.
Okay.
On the Microsoft Store, you're just going to search Ubuntu. And this one right here is going to be fine. This first icon is just says Ubuntu. And you'll just hear for you, it should say install.
I assume I'll need to do the same thing after the changes are applied here.
Exactly. Exact same thing. And so, yeah, I guess we kind of have it fairly easy. So you just press this button, launch after downloads and installs.
Ooh, mine says Windows needs to reboot to use changes. But
so go ahead and reboot then, and you can just join us again after.
Yeah, I'll rejoin you once I have done.
And you already rebooted, right, Batia? Am I saying your name right?
Yeah, you are. Thanks for asking. It's interesting because I installed the Ubuntu, the 20.04 or something, and the only thing that I can find in my directories is the file that I made with Nano, and nothing else shows up.
In your directories.
Right.
So you are using... So do you mean you coded it in sort of this environment like I'm showing on my screen?
I can't see the screen. I have too many things open. Hang on a second.
Oh, okay, that works.
Let's see. See, I've tried so many things. I tried to install it on Unix, on my shell through school, and it wouldn't install QIIME2 because it was too big. And I can't delete anything off of my shell for some reason. There's something that IT did, like make directory and delete, like it just doesn't work. So I don't know. I've got to talk to IT about it with if I'm going to use that shell, but I've been trying to figure out what's going on with Ubuntu, and it doesn't seem to be doing anything.
So in this case, why don't you just try installing this one, the first one. Just do Ubuntu, install
Okay.
that one. Once you have that installed, launch it, and then we'll go from there.
Okay.
And if you have issues doing that, then okay, then we have bigger problems.
Okay.
But it's nothing to be too worried about. We'll just delete everything on your computer. Simple. I'm kidding. Anyway, yeah, wget is an issue for some people on Macs.
Yeah, here's a QIIME's documentation.
Okay,
You just
I'm
kind of
gonna stop sharing my screen, so or you're gonna text it.
have to copy paste these into your terminal.
So I'll stop sharing. And
So like if I have a blank terminal right here, and I just like Control C on this,
so
then I go,
Sam, you're gonna need to actually flip your virtual cam because all the text is backwards.
oh, it showed up on backwards for mine earlier. All right, hold on.
Yeah, and did you enable HD?
Flip horizontal. Okay.
Where's this thing?
No, I'm not sure if I did.
Okay. So right now on your screen, it's very blurry, which is a side effect of, I guess, using virtual cam. I thought it would be higher resolution.
Oh, no, I think it's just my VirtualBox resolution. Sorry.
Oh, okay. So why don't you maybe copy and paste the link to the site so that people can use it.
Yeah, sure.
And Mary Lou is saying to install wget, you type in brew install wget in your terminal. If you have homebrew.
Yeah, this is the one that I was stuck with a while ago because wget doesn't work on my Mac.
Mm.
And so what I did is I downloaded, well, apparently I have homebrew and I didn't know why I have homebrew in my new computer, but it worked. If you have that, just type in brew install wget. Because the ones in Conda, the Conda install wget doesn't work. It needs to be in your user bin.
What about
And
curl? Do you guys have curl?
curl, it works too. I think it should be in the Mac, but I would prefer wget just because QIIME has been doing that. Has been using that.
Yeah, gotcha.
So let me know if you were able to do this. If not, we can. I can, I guess, do a step by step.
We should be able to do something like breakout rooms too if we need to do something like that.
Stefan,
Yeah,
I'm back.
great.
I am downloading Ubuntu right now. So
Okay, you should have it easy once that's up and running.
For those downloading Homebrew, please update your Xcode. Homebrew will not work without updating Xcode.
Steven, I have a question.
Mm-hmm.
For Ubuntu, do I have to install Miniconda in Ubuntu? Because I have it on my computer, but it's not finding it in Ubuntu.
Yeah, that's what I would do. Install it in Ubuntu. So once
Okay.
you've got Ubuntu on, then you're just gonna follow the same steps as Linux, because now you're running Linux.
Oh, okay.
Does that make
That's
sense?
what I'm doing wrong.
Yeah. So you can run that same wget command, but in this case, you're gonna do latest Linux. Let me share my screen.
Linux.
Sorry.
Okay, let me see. You.
So you're gonna run this command wget. I will put it in the chat as well.
Okay.
Did you just say you're posting that into the chat?
Yep.
Awesome.
And then it's very similar to what everybody is doing for Mac, except for you're just using the Linux version, and then you're just gonna run the bash command afterwards.
And it's like a right click to copy or a paste.
That's right. So you would right click. If you're copying into anything, you just right click. But what will happen usually is it'll run the command immediately.
Oh, come on, work.
Yeah, sometimes talking to your computer will help it work.
It does.
So for the Mac people, especially the undergrads, Johanna, Lauren, were you able to install? Are you still in Miniconda installation, or
Yeah, I'm definitely having some trouble trying to install it because it's just like keeps popping up just a screen of like code, I think. And I was trying to do like the homebrew, but I don't have it on my computer and it's just like not letting me. So
Okay, let's just use curl then. Because you have curl and you don't need homebrew.
You can use curl, that's true.
Yeah.
Okay, how do I bring that up on my screen then?
Why don't we let you share the screen, since I think you're not the only person running into those issues.
Okay.
Sound good?
Yeah, that's good. Okay, let's see.
Lauren, I'm going to make you the co-host. Okay. Go ahead and share your screen.
Okay, so like this is what's popping up. Like this, can you see this code? Like when I downloaded that and that's what's popping up. So I just like didn't know what to do with that.
So what you are looking at right now is, I think it's actually the code that's in there. So I think you can close that.
Okay.
And just open that terminal up again. And I think it makes sense to just share your desktop screen if there's nothing too sensitive on there.
No, yeah, that's fine. Like this screen.
Okay, so you downloaded it manually. Got it.
Yeah. Is that not right?
Why don't we try doing it in the terminal, because it'll be only like a 2-button operation.
Okay, where is the terminal?
Okay, share your whole screen if you can.
Is it not? Oh, sorry. Resume share.
Yeah, I'm just seeing. We're seeing like a little clipping of what you downloaded.
Oh, weird.
Can I help Lauren with how to see the terminal?
Sure.
Lauren, go to your let's see, what's that, Spotlight or that Launchpad?
Okay, sure.
Then you'll see other.
Launchpad.
Or you can press Alt space and type terminal.
Yeah, that's true.
Alt. Okay.
Okay, and then do you see other, like disk utility, terminal, desktop monitor?
Let's see, like,
No.
Let's see. Like, should I go under? Because I'm on the Launchpad. Should I go in like Finder or?
No, go other.
I don't see other.
You see it? Go to applications then, and I think it might be in the applications.
Let's do it the easy way, Lauren. Just escape out of all of that. Press Alt and the space key at the same time. It's gonna bring up Spotlight, and then you're gonna just type in terminal.
Got it. Lauren, utilities. That's what it is.
Okay. Yeah,
got-
okay, I see it. I'm on that screen.
Okay, so share that screen, and then why don't we see if we can use curl?
Okay, can you see this?
Yeah, so you're gonna type curl, C-U-R-L, and I'm gonna actually see if I can just copy it into the chat.
Okay.
So you're gonna type curl. I'm moving to the chat now.
Yeah, this is definitely all new to me, so thank you.
So you're gonna type curl, then we've got the input, and then we're gonna put the output as Miniconda latest. Try copying that entire command there.
Okay.
And then to paste it into the terminal, you can delete the curl part since I included it in the thing I sent you.
Okay, let's see. So you said curl and then just like the rest of the website and everything?
With the O. So what you could do is just select all of what I sent you.
There we go. Like that?
Yeah, you'll need a space after the L in curl. And to move there, you're gonna actually have to use your arrow keys.
Okay.
Yeah.
Okay. And then after I put the space, then I just,
And then
okay, there we go.
1 second before you execute that, I want to make sure that you have this other thing right. Yep, that should work.
Cool. And then do I, what,
And then
do I just hit
hit
enter?
return? Yep.
Oh, cool.
So you can see it's downloading the file from the website right now, and it's
Okay.
got it. It says it could not resolve the host.
Hmm.
Okay.
I just tried to do the same thing, Stefan, and I got the same.
You got
mess.
the same issue.
Yeah.
Miniconda 3 latest. Okay. Let's try it one more time. We're gonna go to the Mac. I'm gonna see if it'll get it on my computer.
Should I stop sharing?
Yeah, you don't have to share anymore.
Okay.
I'll see if I can mimic it on my computer to show you, but you're essentially in the
Thank
terminal now,
you.
so that's really good.
Awesome.
So here's my Linux thing, which is more or less running bash. I'm going to copy this command. Let's see if I get the same error. Okay. And I think it is because this might be in the wrong location. No? Okay. Then, what are we saying? Okay. So Jamie, in the chat we're seeing that Jamie just typed in bash miniconda 3 latest.
You would type that into the terminal, or is that like?
I think what you could do is type in bash and then find in your downloads where that file is.
Okay.
And then you just drag that into your terminal.
The Miniconda one?
Yeah, the one that you downloaded. So I think you would type in bash and then drag the file and it'll put the path and then you should just run it.
Okay, I'll try that.
Okay, Michael, I see you say you think you've got it running. That's good. If you do have it running, then you should be able to run this command, conda list. But there's one more step to go through once you guys have installed Miniconda.
Hey, Stefan.
Yeah, Terrell?
Should I run the cleanup? Is that needed? Sorry, underneath install QIIME2 with the Conda environment on the docs, there's an optional cleanup script.
Sure, you can run I don't see there's anything wrong with the cleanup. I think what it's going to do is delete the install file.
Okay, well, sounds good. I'll do it.
Okay.
If I have Miniconda, do I still need wget?
Yes.
Or do I have it already?
I think with Miniconda you should be able to install wget fairly easy.
Okay.
But let's just all get to the Miniconda step and then we can all go together from there because it's the same for everyone.
Sounds good.
So who doesn't have Miniconda going yet? Is it just Lauren so far?
So Stefan, I have Miniconda, but I have it like Miniconda 2. Is there a difference? Should I try getting Miniconda 3?
If you have Miniconda 2, then the update of Miniconda is something that's going to be an easier thing to do. So
Okay.
you don't have to download anything. There's a code that you run, I think, in Miniconda 2. Yeah, Mary Lou just pasted it. Conda update
cool.
Conda. So just run that command and see. Okay, when you guys get to once you've got Miniconda installed, just let me know. You can put a thumbs up and go from
I
there.
think Loren is still having some issues.
I'm installing QIIME2 right now,
Okay.
so it should be done in a minute.
Okay, so you guys are good. Lauren, yeah, just let me know. If Conda list works for you, that means you've got Conda installed, and that's great. So then we will move on to the next step, which is going to be to install BIOCONDA. We already have Miniconda installed on all the computers, so one second.
Question for you, Stefan.
Yeah.
After this meeting, are you going to make this presentation available to us?
Yeah.
Sweet. Thank you.
Yeah. Sorry, it seems as though I'm not finding where the BIOCONDA stuff is. I'm wondering if I put it in the wrong thing. But I have it right here.
Do you want me to link it? Because I can just link the install page.
Yeah, link the install page, everybody, and then
Okay.
BIOCONDA is really simple. You just need Conda, and what it'll do is it'll let you install biomolecular packages much easier. So you just write Conda install whatever package that you want to run, and it'll get it all from the repos and go from there. Okay, great. So go ahead, you guys, and go to that site. You're just going to run these commands. If you're on Linux, you'll run these commands. If you're on a Mac, you'll run the one above, but almost the same. And all you're going to be doing is copying and pasting those commands into your terminal.
Stefan, can you send me that link for the BIOCONDA?
It's in the chat to everybody. Samuel just sent it.
Oh, I see. Let's see. Ah, here we go. Thanks.
Got it. And it looks like actually if you already have Miniconda installed, you don't even need to do these parts. All you need to do is the setup. So you'll just be typing these commands in. But make sure that you follow the same order. So the add channels default has to go first, then BioConda, and then CondaForge.
It might take a little while to install the Forge channel because you have to download a bunch of stuff.
Okay. So you don't have to do the CondaForge if you don't want to, but feel free to if you'd like. And that's it.
One more question.
Yeah.
You have Miniconda. You just say you don't need BioConda, or you do need to download
What
it?
BioConda is it's gonna let you download packages from like this one repository. So you don't have to download the file, but run the commands I put in the text, the conda config file.
Okay.
The conda config, add channels default, add channels BioConda, and then the third one.
Okay, so run those codes. Okay.
If you've already installed QIIME, should I install BioConda as well? Jay, I guess it depends on how you installed QIIME. I'm assuming you might have done like a virtual box, but if you've already got QIIME running, then
So I installed it via, I did the whole Conda or Miniconda thing. So I got the wget and then use wget to get it.
wget. So in that case, just run the conda config once.
The conda config once?
Yeah, in the text, it's right above you. There's conda config, add channels default, add channels BioConda, and then add channels CondaForge. Do you see those?
Sorry.
I'm gonna put it on my screen right here, but it's in the chat right above what you said.
Maybe you have to explain why they need BioConda.
So BioConda is a place where we're gonna be able to download other programs that are useful for doing this kind of work.
Okay.
It isn't 100% required, but I'd recommend it if you're gonna be doing more work along these lines.
For installing that, should I install that in the QIIME environment, or should I do that in like my root environment?
I don't think, you don't need to do CondaForge. I would do it in the root, so I would just exit that terminal, start a new one because it's going to be easy to get in there, and just run these 3 commands. Or 2.
Do we want to keep going on the BioConda website after those 3 commands? If I'm finished with those, the install packages?
No, that's all. That's all you need to do.
Cool. So those 3, the conda config, add channels, conda config, add channels, BioConda, conda config, those 3?
Yep, those 3, and run them in that order, and that'll be all. And that warning sounds fine.
So I got a warning, defaults already in channels list moving to top. That's okay.
That's good.
Okay.
And then you could actually stop at the BioConda if you don't want to do the CondaForge, but
I don't mind.
Yeah, might as well.
Thank you.
Yeah, and the reason why we're doing this is because you're going to want to install other packages later. Like, for example, you might want to work with NCBI Blast, or you might want to work with the cut adapt on its own, or in other sort of configurations. And for that, having BioConda installed will make it very easy for you to install those files.
So Stefan, let me reiterate that. So once you do the CondaForge and it says warning already in channels, it means that you already have it through the Miniconda, right?
Mm-hmm.
It's already been installed before.
Yeah. So, but it is moving it to the highest priority, to the top of the different packages. Okay, and once everybody has done these steps, we can move on and you can actually practice some bash coding, and we'll go back to where I started earlier.
I'm still waiting for QIIME to finish installing.
Okay.
Takes a while.
Did you see the file size that it was?
No.
Okay.
I know it's big though.
Okay, well then while that's happening, why don't we all do the same thing and just download QIIME.
I don't fully understand what's going on in that class right now. Like, if there's like assignments or not, I don't really know what happened yet.
And we are going to be installing it within a Conda environment. So I'm going to send this link to you guys in the chat.
So we had a presentation.
So we can all just go to that link, click it, it'll take you here. For those of you who already have installed QIIME, that's great. Then all you're going to need to do is copy and paste this code for the Mac, like that, and paste it into your terminal and execute it, and it should work. If you're doing it on the Windows subsystem for Linux, you just go to this bit, same idea, run that command. And then if you want to test to see if it works, you'll just run this command conda activate QIIME2 2020.8. And if you want to deactivate it, there's the code right below.
After QIIME2 finishes installing, should I do these Conda configuration add channels defaults to Stephen?
Yeah, I would.
Because I didn't do it first.
I wouldn't do it in the Conda. I wouldn't do it in the QIIME2 environment.
Okay, so make sure I exit
run
out of
conda
that directory.
deactivate. If you
run conda deactivate first.
Yeah, and you'll notice if you're in the Conda environment, it's kind of simple. It's gonna say here. It's gonna say it'll say QIIME2 instead of nothing.
Okay.
So for example, if I activate.
Stefan, before we move on, let's ask the undergrads in our lab and see if they were able to get this correctly, if they were able to install QIIME.
Mm-hmm.
Can you please speak up? Lauren?
Yeah, I got part of it done. I'm still trying to figure out how to do like the conda config, because when I did trying to download the QIIME, it said like command not available or something. So I think I don't have the conda environment set up, so I have to try to do that.
So after you download the conda, just close your terminal and open it up again. That might help.
Mm-hmm.
It's kind of like a restart.
Okay.
So Batya, like I was saying, if you're in the Conda environment, this is going to say QIIME, the QIIME2 environment in Conda. You're going to see this instead of base.
Okay, I gotcha.
Yeah.
Thank you.
And so you want to get out of that back to base.
Okay.
Stefan, question for you. So I am right now running the conda env create command from there. The next step, it says, is an optional cleanup. Would you recommend running that?
Yeah, it's just going to delete the file that it downloaded onto your computer.
Okay, cool. I will run that when this one is done.
Perfect.
The optional cleanup also removes the 2018 version and the 2019 version.
Which I don't have, but would be important if I did, I suppose.
Okay. I mean, in this case, this command right here, Mary Lou, it only deletes this file, which is what you downloaded. There. Okay, I think Lauren, you might be making some progress.
Yeah.
Okay, great.
Hey Stefan, I've gone into a little roadblock. So I was trying to download the thing from BIOCONDA, that link. And it was uploading this entire time, and then it just stopped and said unexpected EOF. So I ignored it and tried to do like the conda config command, and then it's no such file or directory. So I have a feeling that it did not like download onto my computer at all.
So you're on a Mac too, right?
Yeah.
Yeah.
Johanna?
Yeah, Mary Lou, you might need the help there.
Are you using Python 2.7 or and have downloaded Miniconda for Python 2.7, or have you used Miniconda 3 with this one corresponds to Python 3?
I have my Python 2.7, but I'm using Miniconda 3.
Okay, so the Miniconda 3 actually creates an environment for Python 3.8. So you should be fine.
Okay.
The problem is it's not installing properly. Is that what it is?
Yeah, it kind of stopped halfway through. My computer seems like it's overheating a little bit.
It does that when it's busy. Can you show your screen to us?
Yeah.
I'll
Can you make me? Thank you.
Go ahead.
Okay. Cool. Can you see my screen? Cool. So I put in this command, the curl repo anaconda, and then this is it when it was uploading, and then all of a sudden it just like stopped right here.
Yeah. So
and
why
then
don't we download it manually then, just using Chrome or whatever browser you have open right now? Oh, so you ran that command?
Yeah.
Okay. Did you already run the Miniconda installation that we did earlier?
Yeah, so I have.
You already did?
Yeah.
So then just type in, why don't you close your terminal, open it up again? Yeah, the easiest way that I remember searching on a Mac is just the Alt key and the space button at the same time to open your Spotlight.
That's.
Wow.
I think it's command and space bar now.
There you have one right there. Johanna. It's right at Zoom.
Oh yeah, it's right next to the zoom icon on the bottom of your screen.
Go to your icon.
Where?
There. That one. Click that.
Bad.
Okay, so you're gonna wait for it to come on. And then now just type in conda space list. And yeah, looks like you're good. So you don't have to do much now. Now what you'll do is, what did you say the command was? Mary Lou, was it conda, update conda? Update conda. Run that. Okay, that's looking good. You might want to plug in your computer. It scares me. So it looks like good. You've got the latest version.
Yeah.
You are. Okay, now what you're going to do is click on that page, yeah?
So here.
And go down. We're not running that command. We're going to go below to set up the channels. And you can copy all 3 of the conda config. So just all of them at once.
Copy paste all of them.
It's trial by fire.
I know.
And then to paste, you just right click. Or maybe, yeah, you can do that too. Perfect. That's good. That's what we wanted to see.
It's at that beach over there. You could. No, you go over the bridge and then like, yeah, it's not too far. Yeah, past those trees. Yeah, sorry.
No worries.
I was lost.
I think
Is that it?
Hit return one more time. There you go. There you go. So now you are up to speed with everybody else.
Cool, thank you.
Awesome.
Let's stop sharing my screen.
I'll do it for you. Okay, Lauren, how are you doing? Are you almost there?
I think I did it, but I'm not sure because I keep getting weird. Like I did the channels and then it says after like command not found when I'm doing the channels. So
Did you do conda list? Did that work for you?
like type in conda list.
Conda space
Yeah.
list.
Yeah, I did. It says command not found.
Can you share your screen like Johanna did?
Mm-hmm. Okay, so there's this.
Okay. So close your terminal, quit it entirely.
Mm-hmm.
Open it up again.
Okay.
That's fast. And now type conda space list. There you go.
Oh,
So
there we
that's
go.
good. And now you can run the conda config.
Okay. Can I copy it from the chat or do you think that will make it weird?
You should be able to copy it from the chat.
Okay.
Yeah, yeah, it disappears.
Just in conda and config.
Here, this will help you get to the chat easier by having Zoom be normal. Okay.
Okay, I added it.
You got it.
Then,
so you do the default one first.
So I put in what was in the chat, yeah. And then
it should give you like a warning on the default.
Yeah.
And then you run the BIOCONDA one underneath that.
The BIOCONDA?
Yeah, if you ran these 3 commands here, you should be fine. These 3.
Yeah, I ran those 3 commands.
Okay. All right, poof, we did it.
Thank you.
I think with that we can actually try and run some bash coding. We'll just continue with what I was starting at the beginning here. Before I do that, does anybody feel like they want a break, or are we good to keep going?
Is anyone else still waiting for QIIME to finish installing?
I'm still installing QIIME.
I'm using a Mac, not a Mac. Sorry, I used to have a Mac. I have a what's it called? It's like a tablet, but it's not. It's like got a computer environment, but it's touchscreen. Why can't I remember what my computer is called?
And the Microsoft.
Yeah, Microsoft Surface Pro.
Surface Pro.
Surface.
Oh, that's why.
So,
okay, yeah, then Lauren and Johanna, your next step is going to be actually to download QIIME2. So I'll copy this code in there. Did you already do that?
Yeah, I copied pasted it into the terminal, but the terminal says like not like SPEC not found, invalid name. Try.
You did this? You copied this command?
Yeah. Should I just try exiting out of the terminal again?
No, I think you just don't have wget, maybe.
Okay.
Type wget. Type wget. And then
it says no such file or directory.
Okay, then I think you might be able to write conda install wget.
Wget. Okay.
Is it doing something?
Yeah, it's doing something. Thank you.
All right, that's why we want conda, because that's how easy it is to install packages there. You don't need anything else.
Yay. And then, okay, cool. It's doing it. And then after it downloads that, I can enter the QIIME.
Then you should be able to do that. Yeah.
Yay.
Do the conda activate QIIME.
Yeah, once you've downloaded it all.
Conda activate.
So, Batia, do you want to share your screen to show us what you're looking at? And then I think
Sure.
after that, we will take a 5-minute break and come back to this.
Sounds good.
I will stop sharing mine. I will make you a co-host, so now you can share.
There it is. Okay, share screen. This is what I'm looking at. Are you guys able to see the Ubuntu?
Oh, yeah.
And it's just stuck on this one. It's been there for a while.
And yeah, and it's only 5.2 megabytes, so that doesn't make any sense. So
Right.
why don't you just stop that? Just type in or press Control C.
Control C.
So Control C is a way to abort tasks.
Okay.
Great. And now what you want to do is press the up arrow. Let's see.
Okay.
So you were creating an environment.
I was just following the commands.
Try that again.
Okay.
Okay, so now what you're going to do is type in ls.
ls.
And why don't you try conda activate QIIME2-2020? Do you see that up there?
Yeah. Activate. Then, oops. 2-2020. Oops. .8/pi.
No pi. Just delete.
No.
Yeah, there.
Just that?
Just that.
Okay.
Let's see.
All right.
There you go. So that's good enough for now. And if you have any issues, it might be related to the install, but you actually have QIIME. So to test it, just type in QIIME. And then no, you don't need the 2 space -- help. There you go. So now you see it's working, and it tells you all the things. So you could scroll up on that if you wanted to read anything about it. So you look like you've got QIIME installed and you are good.
Okay, great. Then I just need to run those other commands, or after I get out of.
Yeah. So now you'd type conda deactivate, and you could do the conda config ones.
Okay. Perfect. All right, so I can stop sharing.
Yep, already stopped you.
Oh, you did? Okay, you can't tell.
Okay, should we do a quick 5 minutes, you guys, and then come back to it and go through some of the exercises and maybe even go through the tutorial?
Sounds good.
All right.
Wait.
Then I will see you back at 3:07.
Okay, so this is like what it's coming up as, and I've tried to do it like a few times.
Okay, 1 second. What is it saying? Type in ls and hit return. So type the wget command. That you don't have to. No, delete that. Sorry, what they have in your Chrome behind the terminal.
Oh, like this thing?
Yeah. So just copy the first line.
Just the
wget for now. Let's just run that.
Just
no,
this.
the whole line.
Okay.
Yep. did it say?
It says library not loaded. It's requiring like a later version. But
What is happening? Okay, so just copy that error and Google it and see what they say.
okay. Okay.
Okay, well, it is 3:08. We'll get back to just finishing this up. I'll give you guys a brief sort of presentation as to what is happening for QIIME2. So if you have any issues that you can't deal with or can't fix in this session, that's okay. You should be able to work on it over the weekend or next week and hopefully fix some of those errors, like what you were reporting, Lauren. For the most part, it looks like a lot of us got QIIME to go. So here's my It's Alive slide. And of course, you're probably asking yourself, you know, what the heck is QIIME2? And QIIME2 stands for Quantitative Insights into Microbial Ecology. And QIIME2, if you Google it, it's actually a wrapper program. So it's kind of like a burrito shell, like a tortilla. It holds everything inside of it that you can then easily run commands through it. So it's supposed to make things simpler once you get it installed. So wrappers like QIIME2, they have many programs inside of them, like Data2, which is a pipeline to do this sort of analysis that we're going to be doing later. It's got deblur, it's got programs like cut adapt, which will go through sequences and cut out certain sections and remove primers and whatnot. It also has a program called Emperor, which you can use to make these sort of PCA plots here. So this is actually one from their paper. And then you also have added functionality, like you can use other programs or packages that people develop for QIIME2, like PyCrust2 that I've recently found out. Another thing that's great about QIIME2 is it tracks data provenance, which is more or less keeping track of, you know, what steps you had to go through to actually get this file to where it's at. So in this image, you can kind of see that here. We're looking at a folder that's, I think, in the sequences. It tells you how long it took to execute that file. What the action was. Looks like we were denoising 16s. It keeps track of the different parameters that you chose to run the command. And so these are all useful in the future, for example, if you were to make your project available as part of your paper or publication. And what QIIME actually does is that it actually creates these QZA or QZV files. And they're essentially zipped files, so it's like folders in folders that QIIME2 can access and look at through itself. And if you're very confident in coding, like some people are, I'm not in terms of QIIME2, you can actually run most of these commands in Python and work with QIIME2 in that kind of a capacity. And so I'm going to show you how things are easily visualized. This is from a microbiome project that I was working on for my corals that I'm studying. Let me just exit out of here. And all you need to do to visualize something in QIIME is you go to view.qiime2.org, and then you'll see that all you need to do is drag and drop the QZA or QZV file. I'm going to drop the QZV file, which the V stands for visualization. And what I'll do is I'll show you the taxabar plot output. So I'll just drop that here. It loads it. And now you can see that I'm starting to look at the data that I have. And in this case, on the y-axis, we have the relative frequency. And on the x-axis, I have the samples. So I'll increase the width some more. And you can change the different taxonomic levels. So right now I'm at the domain level, but I could go to the phylum level and show that data there, or even further down to the order and show the data in that way as well. And so QIIME2 allows you to do that. I could share this file with you and you could actually visualize this on your end too. And if you had questions about, you know, what I was doing to get there, well, here's a whole map of what occurred to get the plot that I have. And all you need to do is click on it, and it's telling you everything. So this command that was run here, where it went, from there where it went, and so on and so forth. So that's one of the cool things about QIIME2. Okay. Citing QIIME2 is something that they obviously would want you to do. You can cite this author here, or in QIIME, you can just write QIIME citation star.qza. So whatever your QZA file is, or QZV file, if you write this in your QIIME environment, it will tell you all of the packages that went into creating the output that you're using right now. And that can be useful in the future. Another thing you might have noticed is that QIIME2 has an amazing website. It's free, open source, and the community actually develops the program. And that community is really active. So if you go on their forum site, so forum.qiime2.org, they help other people. They're very open to newcomers like myself. They were very positive when I had questions. They also provide tutorials, which we're going to hopefully work through at the end of this. So docs.qiime2.org. And yeah, highly recommend you checking out their site if you want to learn more about QIIME2 on your own time. So I'll just get started. This is from the tutorials overview page, and this is getting oriented to the flowcharts. I edited this a bit to make it more conducive to my computer. And so QIIME2 goes through many different steps. You start with raw sequences, you demultiplex those, join pairs, go on to this next step with these demultiplex sequences, and then actually do the denoise and clustering. So hopefully we're going to get to this denoise and clustering step. But if you look at this right now, it might just be like overwhelming, like, oh my god, too many colors, too many bars. And so I'm just going to try and walk us through this. So let's say we're doing an example experiment. We're interested in looking at the microbiome of a mouse, of a clam in the ocean, a rhinoceros, and maybe something in an aquarium tank. Well, what you're going to do is get those tissue or whatever samples, put them in a vial, extract the DNA, and then amplify the variable for region using PCR, and that eventually gets sent off to be sequenced on a sequencer. Now, a sequencer like this one here, a MiSeq, is plenty. In this case, most options usually use a MiSeq paired end sequencer, so they're doing two sequences of the one DNA fragment that they're looking at, where each sequencing depth is, or sequencing length that they can sequence is 150 base pairs. So if you're interested in doing that in like just a small scale, it costs around $30 per sample, or at least that's what I had to pay for the University of Chicago. But the benefit is if you're doing a lot of samples, the price can decrease substantially. With a single MiSeq run, you can run about 250 samples with 40,000 reads per sample, so that's a good enough quality. And at a cost of $1,641 per lane, if you're running the whole lane on that instrument, you're going to be paying around $6.6 per sample, which is pretty good. It'll probably go up to around $10 because you'll have to do the library step and the DNA extractions, but fairly affordable to get this data. From there, once you've got your samples running on this instrument, it's going to spit out the raw sequences in the format of a FASTQ file. And so this is the first step on our workshop overview of the flowchart. So we're getting our raw sequences. I'm just going to make sure that you guys can hear me real quick. Share computer sound. And I'm going to introduce you to one of my favorite YouTube channels on the internet called StatQuest. Be prepared. He's kind of slow, but he has a really great way of explaining things. I'm going to let him speak, and I'm going to speed up his speed to about 1.5, and you can let me know if that bothers you.
Hooray! Now we sequence the library. Let's see how this is done. Imagine this is a fragment of DNA we want to sequence. It's vertical because that's how it is inside the sequencer. Actually, There are about 400 million fragments laid out vertically in a grid. I'm just showing you 4 fragments so your brain doesn't explode.
Sorry.
Just showing you 4 fragments so your brain doesn't explode. This grid is called a flow cell. The machine has fluorescent probes that are color-coded according to the type of nucleotide they can bind to. The probes are attached to the first base in each sequence. Once the probes have attached, the machine takes a picture of the flow cell from above that looks like this. The picture tells the machine that the first base in the bottom left-hand corner is an A. This base is a G, and these 2 bases are C. Then the machine washes the color off the probes. Then the probes are bound to the next base in each fragment. The machine takes a picture from above, and now it knows that this base is C, this base is G, and these 2 bases are T. Then the machine washes the color off the probes, and the process repeats until the machine has determined each sequence of nucleotides. This is how it works with 4 DNA fragments. With 400 million DNA fragments, the matrix is much denser. This matrix still isn't 400 million DNA fragments, but it illustrates one type of problem that can occur. Sometimes a probe will not shine as bright as it should, and the machine isn't super confident that it is calling the correct color. Quality scores that are part of the output reflect how confident the machine is that it correctly called a base. In this case, the faded dot would get a low quality score. Another reason you might get a low quality score is when there are lots of probes the same color in the same region. This is called low diversity, and the overabundance of a single color can make it hard to identify the individual sequences. The colors will blur together. Low diversity is especially a problem when the first few nucleotides are sequenced, because that is when the machine determines where the DNA fragments are located on the grid. Now the 4D for the sequence.
Okay, so it showed you more or less how it gets the data, how it figures out what the different nucleotides are by using essentially camera to do an image of the flow cell. It shows you that there can be some issues, so the instrument has to know or more or less give you an estimate about how certain it is that it's making the right call. And that's why it creates something called a FASTQ file, where the Q in FAST stands for quality. And I'm just going to go through what that looks like in terms of output. So here I have a table where I'm listing the function of the actual file that we're going to look at, and then the value that it will show you. So the first thing it's going to do is show you the sequence ID, which you can see here, the sequence ID. And this is just a unique identifier that identifies a specific sequence. Below that, it's going to have the nucleotides that are corresponding to that sequence. So this is what's going to identify the sequence. And it's later what we're going to use to sort of figure out what kind of microbe this might come from. After that, there's a separator, which is just a plus sign. They're not entirely sure why that plus sign exists, but it's essentially going to help separate the nucleotides from the quality scores which come below. And so this is where the instrument is telling you how confident it is in the quality of the read. And in general, if you're seeing a letter, that's a good sign that you have high quality data, although we're going to run programs later that are going to go through this step all on their own, so you won't even need to interact with this kind of a file type. But it's good to know. And then below the quality score, we're going to get to our next sequence ID. All right, so we know that there's quality scores. They're different letters. They're essentially ASCII characters. What did those quality scores mean? So quality scores are been into 4 different groups. A quality score of 10 means that the error probability is about 1 in 10. Going up from there in orders of magnitude, now it's 1 in 100, 1 in 1,000 for 30, and 1 in 10,000 for 40. And this image in the background is showing what the different characters mean and what they correspond to in terms of the Q score. So you can see here that A is an ASCII code of 65 and it corresponds to a Q score of 32, which would mean that it's a pretty good error probability. This is going to be a good read for your machine. And so in general, 10 is bad, 20 is questionable, anywhere up from 30 to 40, that's going to be really high quality range. And what you can see from our actual fragment that I showed you is most of the nucleotides that it told us are really high quality. There's just one, which in this case was this dash, that was a lower quality score, and that might need to be filtered out. So a FASTQ file contains that information, and that's important for many different processes following this step. So our sequencing instrument has created the file, the FASTQ file. It then sends it to your computer. Your computer does the processing, identifies these different nucleotides, and you can figure out what bacteria are in them, and it can create a FASTQ file from these. Again, another one. But how do you actually go to figuring out which organism these samples came from, or these sequences came from? And for that, there's something called multiplexing, because the way we have it right now is it wouldn't be able to tell you anything about where those sequences came from. You can't figure out which one of these fragments came from the mouse or the rhinoceros, for example. So multiplexing uses barcodes that attach to either side of the DNA of interest and serve as a unique identifier. So for all of the mouse genes all of the mouse microbes that we amplify, we can give them all the same barcode at the end and repeat the same process for the other organisms. That way, when we run them in the actual instrument, we can tell them apart later. So here we're attaching the barcodes to all of the 16S DNA from the mouse, and from the shell, the rhinoceros, and the fish or the aquarium. And now we sequence that, doing those same steps. And now you can see that it's actually going to be fairly easy for a computer program to tell which sequences came from the mouse. And to do that, we go through the step of something called demultiplexing, which is part of the workflow as well. And from that, we can separate all of the data that was in one flow and split it to the 4 different organisms that we were interested in. And so this brings us to the second step now. We're at the demultiplexing step. So to demultiplex, let's say you send off your sequences to a facility. They give you back this one huge file. If you do the demultiplexing step yourself, you might be able to save some money, so it's good to know these things. Although most companies will demultiplex it for you, and so you'll have already demultiplexed reads in your inbox. But in any case, you're going to need a metadata file. This is an example of a metadata file. The first column is going to have our sample ID, so this is what we're calling each of the different samples in our repository or in our data set. Underneath that, you're going to have the variable that's associated with these values. So in this case, it's just Q2-types. So these are the sample types. You'll have the barcode sequence. This is essentially the barcode we put earlier to multiplex our sample. That's what this is telling us. So our metadata is telling us that our samples have not been demultiplexed yet, and we'll go through steps here of actually demultiplexing them. And you can see that it is given this a category called data value. And then you can actually put other information like, the site that this sample came from. In this case, the first 2 came from the gut. You can see there's some from the left palm of somebody's hand. And you can just keep adding as much metadata as you want later for doing your statistical analysis or creating different plots that you might be interested in. And so this is what the metadata file looks like. And then if you did paired end sequencing, you'll want to actually join the pairs. So I'll just explain fairly briefly what paired end sequencing is. So earlier I was saying that in a MySeq platform, you can do a 2 × 150 base pairs. So let's say you amplified the variable for region of 16S rRNA. What you're going to do is you're going to have a forward read that looks at 1/2 or a little bit more than 1/2 of the V4 region. And then you'll have a reverse read that goes in the opposite direction, which I'm trying to capture here by reversing the letters. And what this means is that in the middle, there's going to be a section of overlap, which can actually improve the quality of your reads and how certain you are that you have the right base pairs for this region. And so what most sequencing instruments will do is they're going to spit out 2 unique file names for the reverse and forward reads. So in QIIME2, what you can do is then go through the opposite process of combining those 2 reads into one to figure out what the variable for a region is and what all the nucleotides in it are. And once you've done that, you have demultiplexed the sequences by joining them. fairly simple step, even just logically thinking about it. And from there, the next step is to do the denoise and clustering. But I was surprised to see that they didn't actually say that there actually needs to be a quality control step between these two. Now in the quality control step, what we're going to do is trim our sequences. So remember the quality scores we were talking about earlier. There can be instances where the quality is low in the beginning, so that you might have some reads in the beginning that have quality scores in the region of around 20 here. And it's generally low towards the ends of the reads, which you can see with this graph here. Whereas we increase in the base, so starting from 0 to around 130, the quality score starts to drop. And so what you'll generally have to do is make a call as to where you split, where you decide to trim this fragment. Which I think I might have sent you guys a paper on that, but there was one that discusses what different settings to use. Now luckily for you, the default version that QIIME2 uses is perfectly fine for most cases. For other ones, you might want to adjust the settings by different runs, and generally you do that by having your mock bacterial data set as a part of your sequencing steps. Yeah, it's a lot, so we won't go into this, but just know that there's a quality trimming step. It's fairly simple, fairly straightforward. You're just getting rid of bad areas. You're cutting down the length of the fragment so you have good quality data. And the default settings that QIIME2 uses are going to be perfectly fine. If you want to read more about what's all involved in this, you can check out the Bokulich et al. 2013 paper, or just take a screen grab of what you're seeing here and you can just look through that. And if you want me to cover it in some more detail, I'm happy to do that as well. But essentially, you're just cleaning it up and using a very standard frame of doing that. And then we get to the denoise and decluster section. The old way of doing this, and so essentially what we're doing is we're finding the sequences that are similar to each other, finding similar sequences, and grouping them in some way. And what they used to do is use operational taxonomic units, or OTUs. And what they would do is they'd have these different sequences, and they say, okay, if you're 97% similar to each other, then we're saying that you are a taxonomic unit, and we're going to use you to do our statistical comparisons in terms of alpha and beta diversity. That was okay back then, but now we've moved away from that, and we're going to use amplicon sequence variants, which cluster fragments by up to 99% similarity. So this is actually better because it allows you to tease apart really minute differences. I think the Diebler or the Data 2 paper was discussing that. They had some samples where they were looking at microbial DNA from vaginal samples, and this is usually an area that is super, what's the opposite of low diversity, so there's usually only one genus of bacteria in these samples. And by using these amplicon sequencing variants and Data 2 and Diebler packages, they were actually able to find that, oh, there's actually some minor variation in the sequences from these samples, and they found 6 different groups which might be of interest for future studies. So having going up to 99% similarity is going to be better and help you tease apart some really minor differences. So with that, I'm pretty much done with showing you the basics of what's going to be happening, and we can move on to actually trying some of the tutorials. Before I do that, does anybody have any questions?
Let me specify about Data 2 and OTU. So for ASV normally, if you want to really look at diversity, because that's the variants, for instance, really show you how diverse your community is. But it doesn't mean that it's diverse in function. So there's something you need to actually think about too. So OTU group them by genus level. So this is a conservative way of looking at, well, trying to deduce their functions. For instance, basically what you'd say is, all of these bacteria that have this genera have the same function. On the other hand, that may be different in some environment. And so this is where this is where ASV comes in, especially in an environment where there's very, very low biodiversity. But then again, it's all about function sometimes.
And you can still do the functional analysis of ASV. So right now, at least most of the people that have been developing the packages for QIIME2, they say they're moving away from OTUs and they're going to ASVs. And ASVs are pretty much very similar to OTUs, except for we're grouping different sequences at 99% similarity instead of 97%. And this is actually pretty doable because the quality in the reads is fairly high. Okay. So, what time do we have? We've got 3:33, so we can actually go through the moving pictures tutorial if you guys would like. I think that would be a good use of time. So we're going to go, feel free to follow along by actually going to this page that I've got on the slide here. So docs, I'll put this in the chat. Docs.QIIME2. This is the moving pictures tutorial. To do this, the first thing that we're going to actually want to do is run the code conda activate QIIME2 2020.8. I'm also going to put that in the chat, and I will be doing it on my computer. And there's more tutorials on the QIIME2 website, so if you finish this one, you can go and try some other ones on your own time. And it's fairly straightforward, so they do a great job of explaining everything. So we activate our QIIME2 environment. I'm going to go here onto this, split my screen. And now what they want us to do is run this code makedir, which says make directory. And please let me know if I'm going too fast for you guys. So make directory, QIIME2 moving pictures tutorial. I've done that. Now to actually see what's happened, I can type in ls, which essentially is a shortcut for list. So list everything that's there. And you can see I have 3 different objects coming up. I've got Miniconda 3 latest, which is what I was downloading earlier. Here's my Miniconda 3 that I installed. And now here's that new directory that I created, the QIIME2 moving pictures tutorial. So now what we're going to do is we're going to run the next command, which is cd, which stands for change directory. And I'm going to show you a little trick here. Actually, which you guys probably have not done yet, but for me I've done it. So I can type in cd and start typing QIIME and then just press tab and it'll auto-complete the rest of my line. And so now I'm in the QIIME2 moving pictures tutorial directory. And so it's essentially just like navigating with your visual sort of GUI, you know, clicking into a new folder. But in this case, we're doing it in the non-visual way. Okay, and so now we're going to download the metadata file. You can use wget or you can use curl. I think everybody should have wget. So I'm just going to do that. Press this icon to actually copy the code. And what wget stands for is webget. And we've got these little dashes in the code, and what those do is it allows you to split this code over multiple lines. And your terminal will actually understand that it's saying, hey, this is part of the same command, this is not a new command. So that's what those backslashes do. So I'll run that. It's got my data, and to actually see what I have, I can type in ls again. You can see that my directory that was empty before actually now has a file in it, sample data sample metadata.tsv, and TSV in this case stands for tab separated values. And if I wanted to, I could look at what this file actually contains by writing a very simple command, which is head, and then putting in the name of that file. So sample/metadata.tsv. And now here I see, okay, here's what that file contains. All of my metadata information. Yeah, the next step is going to be to the make directory. What's that?
For those that want to visualize that TSV file, you can open it using your Excel file or numbers, for instance. But yeah, you can. I mean, basically what you did is showing it all. But this is for students that don't know where those files are. Where is the make directory? Where did it bring it to?
Yeah, and actually that brings me to a point for Batia and Michael. Right now, the environment that you're in Ubuntu, it's not the place that you can actually access by using Finder. So to get to a place where you can access Finder, I put this in the Slack page, but there's a command that you can write that essentially is win home, and you can see this this code that is pasted here. I'll put that in the chat. So it might be good for you to actually change your directory, go to the users area, and then sort of get out of there. So when I've done mount C user Stefan Bitterwolf or Stefan, that is actually going to take me here to a place that I can find on my computer. So that's important for you guys to know. Everybody working on a Mac, I'm not sure if it's as important. So what you'd want to do is type in cd, change directory, mount C users, and after users, you'll want to actually put your user name.
Okay. So instead of make directory,
do you see the chat?
Yeah, mount C users. Okay, CD.
Yeah, so just
CD
copy.
mount C users. Okay.
You can copy that, and then.
Okay, so I'm in there.
And then type in CD again.
Okay.
And put your username, whatever it is. You can type in ls to see what your username is if you're in users. ls lists all the things. You can see all users default user public, and Stefan. And that would be my username.
Yeah. Okay, so I think I'm in it.
Okay, so I would just do, so for example, if I'm here where you were, I would go cd, type in my name, Stefan. And now I'm in the Stefan user directory, which you can see is full of a bunch of stuff that I can see on my computer. So now just briefly, I will, yeah.
When I do that, I get an error that there's no such file or directory.
When you did mount C users?
Yeah.
Did you ls after it to see what your username was? Because mine was different than what I thought it was.
It won't let me into either just users with no name after it or with my username added.
Let me just open Ubuntu up again. Try closing it, Mike, and just opening Ubuntu up again and starting from there. It might be that you're just further up or something, and that's affected.
That's fair.
What you can do. So if you're in base, type in ls. See if you can cd to mount, which I can. So just see if you can cd to mount, and from there you can type ls again, and now I see all my different drives. So now I can cd to c, change
Just
directory.
cd/mnt/. Is that
Yes.
the?
Or you can just do cd mnt and then step by step go up. Like I'm doing on my screen, you see I did cd to the mount. Then I saw all the drives that are there.
I think I found the problem.
Okay.
Mine requires a space between cd and /mnt.
Yeah, I'm sorry, was there no space?
There was no space when I copied it.
Okay. Yeah,
Cool.
there's this. Okay, so now you should be able to do it. And so for me, there's a way to sort of hotkey that command to just one thing. I just type in win home and now I'm home. The video that I shared on the Slack will help you do that. And there's ways of doing a similar thing for Mac if you're interested in. So just to sort of speed through this, I'm going to do the same thing. Looks like, yeah, I'm in QIIME 2 moving pictures. I'm going to get my data. And now here I am. I'm going to go to my users. I'm going to look for the actual file that I was looking for, which we called the QIIME 2 tutorial. Here it is. And now I can see that I've got all this stuff. Delete that. So here's my sample metadata. If you wanted to open that in Excel, you can. The best way to do that is to make a new thing, open this up as a text document, like I've got here, select all of it, paste it into one row. Now we've got all that information, select it all, double click, and you can see it all separated fairly nicely here. And so there's your metadata file that came with this tutorial. So we've got our sample metadata. We're going to go from there and copy this next command to make the directory EMP single end sequences. We've made that directory, and now we're going to download our files, the EMP single, and what you can see here is that they've coded in this -o EMP single end sequences with this dash, and what that's instructing wget to do is to download this file below and actually put it into the folder above. Because right now you can see that we're not in the EMP single end sequences directory. We're in a directory one step below that. So when I press ls after this download is done, you're going to see that it actually doesn't find this file that I actually downloaded, the barcodes. So to see the barcodes, I would have to change the directories into that EMP folder, and then sure enough, there it is, barcodes.fastq.gz. But the tutorial has another download step, so we don't need to change directory there yet. We'll copy this next code, which is downloading the sequences.fastq file. So that was 24 megabytes, and I've got that downloaded. And now we have pretty much all the data that we need to start with our first QIIME command.
Just a reminder that this is a single end sequencing. So it's not a paired end. So normally what you would get from Illumina are paired ends. So just FYI, okay?
Thanks, Mary Lou.
Stephen, I'm sorry, but I'm kind of stuck with the command for when home didn't do anything.
Yeah, it won't do anything for you because you have to set that up. But if you just, I think earlier we CD'd into mount users and then we put your name.
So I'm still there. So then I was
perfect.
just supposed to keep going from there, doing the make directory and single end sequences command?
Yes. So instead
Okay.
of my username, your username should be there, and then you can just follow along from there.
Okay, perfect.
Yep. So it shouldn't take you much time to catch up. Okay, yeah, so like Mary Lou was saying, this is all single end sequences. This is what they wanted to start us out with. There's other tutorials after this which will use paired end sequences, so there'll be a different command for that, but also very easy to follow along with. So now we're going to get to our first actual QIIME command, which is specifically using QIIME2 to do the work. And what it's going to do is it's going to use the program that it has called tools, and it's going to tell tools to import. The import you see has 3 different input criteria. The first is you need to tell it what type of input you're specifying. So here you can see we're telling it, this is an EMP single end sequences. And that's good because it tells it what to look for in terms of importing this. If it was paired end, it would look for different data in your metadata file to do the separation. Then you tell it the input path. So we're saying that input is actually stored in the EMP single end sequences directory, which is 1 step above where I am now. And then we're going to tell it the output path. So it's going to take that input, it's going to generate a QZA file, and it's going to put that back into that same directory. It should be in the same directory, and it's going to end it with a .qza. So I'm going to copy that. I'm going to run it here. Copy, paste, with just a right click. I'm running it. As you can see, I'm in base. I did not activate my environment yet. So if you get that error, command QIIME not found, that just means you did not activate your Conda environment for QIIME.
Yeah, I fixed it before he got to it, but I definitely got that error.
You got the same one. So here we go, QIIME2 20. Now we're there. Now I can copy this command and it's gonna work. I'm so confident it's gonna work. So it might take a little while. That's normal. If you do want to sort of get a guesstimate of how fast things are going to go, you can always type the command time before any of your terminal commands, and it'll actually tell you how long it'll take to do a certain thing.
Sorry again, I think I missed something that you did after. Let's see. It's hard because I have to keep looking back at the other screen.
I know there's a big learning curve, but I'm confident you'll get there.
Okay, so I made the directory, and then I used the wget or wget. And then I did that again with single end sequences fastq. And then I did the QIIME tools import. And that seems to be not doing anything.
Seems to be not doing anything, or is it waiting?
Oh, it did stop doing something. Okay, I think it was just waiting.
Yeah, it just takes a while. And that's because this is using only one CPU on your computer, so it's fairly slow when it does these things because it's zipping and unzipping many files and putting them in one place. And I think what that's supposed to do is optimize memory usage to reduce how much memory it needs.
Okay.
So it makes it, broader and you can use it on many more computers, but it does take more time.
Okay.
Okay. So now we should have an output artifact.
So I have I got an error.
Yeah, Jay. What did you get?
So it says there was a problem importing EMP single end sequences, unrecognized file.
Unrecognized file. And do you see that if you're in a QIIME2 2020.8 environment? Do you see what I see?
Yes, I am in the correct environment.
And are you in the QIIME2 moving pictures tutorial directory?
Oh shoot. I think I was. I just accidentally changed out of it.
So if you type ls, you should see what I'm seeing. What you would see is just the EMP single end folder and then the sample metadata.tsv.
Okay, let me try and run it again.
Okay. And once your command is done running, you're going to get this file here, emp single end sequences.qza, and that was the output. So when I was explaining this earlier, I wasn't sure where this was going to go, but sure enough, it is going to output this wherever you currently are. And because I'm currently in the QIIME2 moving pictures tutorial directory, or folder if you want to call it that, that's where it ended up putting it.
So I did just get the same error again. I am currently in the moving pictures tutorial. There's two files in here, the EMP single end sequences and then the sample metadata TSV.
Okay. So let's change directory into the EMP single end sequences folder. And
so in there I have the barcodes, FASTQ, gzip, sample metadata TSV, and the sequences.fastq gzip.
Let me just make sure you see this. So you've got barcodes.fastq.gz and sequences.fastq.gz. Yeah?
Yep. If you want, I can just share the screen with you.
Yeah, why don't you share your screen?
Directory, because it might be on the moving picture tutorial. Oh, sorry, it might be on the importing tutorial, QIIME2 importing tutorial, especially if you're using Mac.
Go ahead, Jay. You can share your screen.
Yeah.
Let's see what he's working with and what he's seeing on his side. And hopefully that will help. Okay.
Sure.
Okay, you've got that. So now what you're gonna, so you cd back, cd space ... like you did earlier, or are you in the?
I was in the moving pictures tutorial.
Okay, sorry. Go back to the moving pictures tutorial. Yeah. Okay, and when you run the command here, you get an error. There was a problem importing EMP single end.
Yeah, if I do that.
Did you by any chance open up that sample metadata file and save it in a different way?
No.
Okay.
Let's see.
Why don't you just delete that EMP single end folder and
try it over again.
Yeah, just start over and see if that fixes it.
That's the right command, right?
You're going to do remove, and then after remove, you're going to put a -rf.
Is it capital R? Or?
No, all lowercase.
Just R.
What that does is it'll delete the folder and everything in it.
Okay.
So make sure you are doing that on the right folder when you do it, because it does not ask for permission afterwards. It could delete everything on your computer.
Gotcha. Should I delete the sample metadata as well?
Yeah, let's do that, because that seems to be what it's having a problem with.
Do I need to do RF as well, or just?
You do not. So when it's just one file, it's easy.
Okay. So Where are we?
You've already got the directory, so that's good. And all you need to do is download the sample data.
Or do I need to? I do need to make the directory, right?
First, download the metadata.
Right.
So you can just copy. See the little clipboard next to it?
Yeah. This Zoom chat is getting in the way.
Yeah. Okay, there it is.
So then we're going to make the directory.
Perfect.
Cool.
And then copy those two. And there's one below it. And for you guys, that extension .gz, that stands for gzip, which means it's a zipped file. And QIIME2 can actually work with zipped files, but when you do this on your own, it usually doesn't. You'd have to unzip those files first. Okay, that looks good. So then just cd one level down and go ahead and try that command again. All right,
cool
great.
Beans. Thank you.
Awesome. Okay, so that's good. So if an error like that happens, maybe it's just something we just accidentally changed it. You are welcome to run this command here, QIIME tools peek EMP single end sequences. So that's going to just give us a little header as to what that file has. It's telling us the ID, what type it is, the data format, and that can be useful, but I've never run it. And that brings us to the next step. This is the demultiplexing the sequences step.
Stefan.
Yeah.
If I might interrupt for just a second. So I ran that peek.
Yeah.
I got something different from what it shows. The type is the same, the data format is the same, but the UUID is different. Is that a problem?
No, that's fine. Mine's different too. It's just the unique ID that it's giving on your computer. Maybe as part of the tracking system.
I did that and it gave me an error.
Okay.
So I'm not sure why, but it says there is a problem with the command invalid value for artifact visualization file. EMP single end sequences.qza does not exist, but I'm in that directory right now.
So you're in it, and when you press ls, you see it.
Yeah, when I press ls, it tells me that I have the barcodes fastq.gz and the sequences fastq.gz.
Okay, so now you need to cd down one level. Cd space ...
cd space ... Okay.
And that should, and now do ls, and you should see the metadata file.
So when I do ls, it shows me like all of my files on my computer.
Okay, so then then you were in the right directory, and I thought you were in one level above.
Okay.
So go back to where you were earlier.
CD. Oops.
To the QIIME2. And you guys, it is 4 o'clock. If you want, we can keep going through this step and get to the end. I think it's too much longer. Or we can at least finish the demultiplexing sequences bit, which is simply, for those of you for who it is working, just go ahead and copy this and paste it in.
Okay, so
and then Batia, we'll cover yours.
Which directory do I go in?
You're gonna want to be in the QIIME2 moving pictures tutorial directory.
Okay, CD.
And I wonder if tab completion will work for you. I'm I'm not sure.
I was trying it, it didn't do it.
Okay,
But I
so
might.
I think we might end on the how to set that up. It's going to be very helpful. Okay. So once you're in the directory, let me know, and we'll go from there.
Time to single end sequences.
Time
So nothing's.
to dash moving pictures tutorial.
Oh, sorry.
Put it in the chat.
Time to dash moving pictures tutorial. Thank you. You're awesome, by the way. This has been really great.
No problem. I'm glad. So once you're in there, you should only see one folder, which is the EMP single end sequences, the metadata file, the sample-metadata.tsv, and then if your previous command worked, the one where we actually told QIIME to import the data, then you should see one file, the emp-single end sequences.qza.
I might have missed something somewhere because it says I don't have the moving pictures tutorial. It says no such file or directory.
Go in the chat and copy the thing that I just sent to everyone.
I did.
You did, and you don't have that. Okay.
Yeah, I did that because it's faster than me typing it.
In that case, just start from the beginning and do
Just
it all
start
again.
over. Okay.
Yeah. Or you just need to
Okay.
find where that folder is on your computer, but at this point, that might be the easiest.
Okay.
So I was having, when I was executing that script, I was having some problems, like warning and stuff, and it could not find a TSV file. But what I did is I put them all on the moving picture and folder, and I just CD on that, and it worked.
Okay, so I wonder if I'll get the same issue. So I'm copying this whole command, I'm pasting it in. This is where we're demultiplexing. And just to sort of explain what this command is doing, so it's saying, hey QIIME, run the demux file. We're going to do it for EMP single format. Here's our input. Our inputs are sequences, and they are stored in the EMP single end sequences.qza file, which was just made. The metadata is for barcodes and file is stored in the samples metadata.tsv file. And the metadata for barcodes are in a column, and that column is called barcode sequence. So if I go and show you that previous thing, here's that column, barcode sequence, and here's all the barcodes that it's looking for to create different files or different information for each sample. Okay, and then it's going to say, hey, output, we want to per sample sequences, call it demux.qza, and then output the error correction details just so that we have them. We're not going to do anything with them, and we're going to call those demux details.qza. So mine ran successfully. We've got it all here. If I type ls, I'm going to actually see that. So here's my demux details. That's where it has all the error correction information. And then here's the demux file.qza. And once again, QZA is an artifact, so you can't really, it's not something that's user-friendly even to look at. Anything that we can look at is always going to be a QZV extension. So this final code here, QIIME demux summarize, that is going to actually give us something to visualize this output. And I think once we get that visualization, we can end. So QIIME demux summarize, so say, hey QIIME, do demux. Now we're going to actually summarize. Here's the input data, it's that demux.qza file. And we want you to output a visualization and call it demux.qzv. So we'll just run that. Should be fairly simple and quick. And then to see the output of this file, I'm going to make a new tab. I'm going to go to view.QIIME2.org. I'm going to go to where this folder, so here you see we actually created it. I'm going to go there on my computer. So here's I'm already there in the directory, QIIME2 Moving Tutorials. Here's my QZV file that I'm looking for, so QZ. The V is for visualization, and now I can visualize it. And it's giving me all of the different data or information about our samples. So for example, it has the sample ID. It tells me how many sequences that sample has. So we've got almost 19,000 sequences in this file. And then it does that for all of the other files below it. And you can see that for some of them, we only have 1,854 sequences. And that can be a quality control issue later on, where we'll want to make sure that these are all subset to the lowest value that we can tolerate. So sometimes you'll have samples that don't have enough sequences, so you'll have to discard them. In other cases, you have enough sequences, but you want to make sure that all of the samples have the same amount, so you'll do like a randomized subset to make sure that you all have the same sequence count information. And up at the top here, you see the interactive quality plot. And now we can actually see how the quality score looks for our data, and we can start making calls as to where we might want to trim it for the future ASV or clustering steps. And so this is an interactive plot, so you can click on one of these, and it's going to tell you the position. You see the position right here, that was position. So here's position 115. It's telling me data for that position. It's saying that the middle of the box, or the median score there, is 29. So that's a fairly good quality score. That's okay. And then up above that is 33. But we will want to start making a trimming, start to trim some of these sequences. And it's kind of subjective, and it will depend, but in this example, we're going to trim it at around 120. And you can see up there that little black line. I think that's the average. Yeah. And for those of you who are having trouble with getting any of this data to work on your computer, and you want to see what I'm seeing, you can also, on the tutorial, press the view next to it. Or you can even download the file instead, up to you. So if I press view, it's going to take me to that exact same sort of thing. It might be a little different because, yeah, who knows why. No, in this case, it shouldn't be any different between my computer and the original one. But there are some places where it would be a little bit different because it's doing a random sampling of that total. Okay. Yeah, so we're going to stop there if you guys want, and we can continue next week, or you can go on your own to look more at sequence quality control and feature table construction. Also, fairly simple way to go about it, just copying and pasting. And then Batya, if you want help making sure that it works on your computer before I go, I'm happy to do that too.
Oh, thank you. That would be great.
Okay,
Not
so
sure what
let's
I
do
did
that
wrong.
after everybody goes. Does anybody have questions before we leave?
One quick question.
Yeah.
So I'm unable to open the demux file, or at least I don't know which, or how do I get the visualization?
So that's correct. You cannot look at this on your computer. You need to open a browser and go to view.qiime2.org. And then you're going to take that demux.qzb file and drag and drop it into this area here. And it's going to do the visualization for you.
Just FYI, for mine, it did not work on Firefox, but it works on Chrome.
Okay.
Okay,
That's
good to know.
Thank you, Stefan.
Yeah, thank you guys. Thanks for being
Yeah,
here.
thanks a bunch, Stefan.
I hope that
Thank
was
you.
Thank you.
helpful.
Thank you so much.
Very patient. Thank you very much, Stefan.
Thank you.
Awesome.
Sweet,
Bye.
it works. Thank you.
Great, Jay.
Have a good weekend.
Yeah, you too. And I'm gonna stop recording and I will help you out, Batya.
Okay, thank you.